Good afternoon, everybody. I'm Dr. Scott Friedman. I'm the Dean for Therapeutic Discovery, and I'm absolutely delighted to introduce and MC this year's Mount Sinai Innovation Award ceremony, which, owing to the pandemic, is on Zoom again. This is the 6th year for this ceremony. Each year it gets bigger and better, and really was born out of the vision of Dean Charney, initially when he appointed me in 2013 as Dean. Whereupon we started the Sinai Innovations event in 2013, and that's been followed since 2015 by this award ceremony, which recognizes the extraordinary achievements in innovation and entrepreneurship across our campus and our community. And it's really Dean Charney's vision that we both encourage, but also recognize the talent of so many people around the Mount Sinai community and campus. It's been really inspiring to see what a wellspring of creativity has we witnessed this year and the last 2 years really during the pandemic. Not necessarily in spite of the pandemic, but it's driven a higher level of creativity, resilience, persistence, and entrepreneurship by all of those who are being recognized today and many others like them. And so it's very fitting that we encapsulate these accomplishments by this award ceremony, which will, as I said, recognize a range of talented people throughout the campus. With that, I want to introduce, actually, can I just get the next slide first? I want to acknowledge the event hosts, not shown on this slide, but very important for the logistics and the planning has been Jacqueline Laws, Najib Abdullah, in Rama Iyengar's team. But in addition, this really represents a team effort on the part of all the individual and groups you see listed here: Mount Sinai Innovation Partners, the Office of Faculty Development, the Conduits Institute for Translational Science, Sinai Biodesign, our graduate school, Department of Medical Education, Office of Postdoctoral Affairs, the GME office, as well as Killen, which you'll hear more from Ilse Donovan, which is the keystone for incubating innovation for life sciences network. With that, I'd like to introduce our Dean, Dennis Charney, who is really responsible for creating and projecting the vision of innovation that really underlies this event. Dean Charney.
Thank you, Scott. But you're giving me too much credit here. That all the work that you do and the team that you just recognized is critical to the accomplishments that you're going to hear about today. I've said this before, that in the age of COVID, the adversity associated with COVID, it reveals character. Not only character in the usual sense, but the character of our scientists and their devotion to making discoveries that make a difference and to our patients. And that was never more evident than what's happened over the last 18 months. And it makes me very proud to be the dean of so many great students and faculty that are pushing the envelope of science, taking risks to make those discoveries that are so important. It takes a village to do this. We've been helped over the last year or so by donations from many generous benefactors that helped us during the beginning phase of the pandemic when we were the epicenter of the epicenter to do the kind of science that you're going to hear today in terms of understanding the disease associated with COVID, developing vaccines, and so forth. So it's really an incredible story. You can hear part of that story today. And in fact, we've written a book about it that is going to be published in the next few months that tells the story of what Mount Sinai did under the incredible stress of their pandemic. And the title of the book is Relentless, which I think characterizes what the scientists here today, the students and the faculty have accomplished. So congratulations. And I look forward to hearing what you all have done over the past 16 or 18 months. Congratulations.
Thank you, Dennis. We'll turn now to the prizes, and Dean Charney, we'd love for you to stay as long as you can. The first prize is to be awarded by Eric Liam, who's the president of Mount Sinai Innovation Partners and the Chief Commercial Innovation Officer of the Mount Sinai Health System. I'm thrilled to introduce Eric, in part because he joined us about 6 years ago from UCSF and has been a real game changer and an impact player in terms of creating a partnering team that really helps educate faculty, also establishes opportunities for commercial transactions, and has really executed on some very big deals that maximize the full commercial potential of the talent that you're hearing about today. So Eric will be introducing the Inventor of the Year. Eric, want to take over?
Great, thanks, Scott. I appreciate the kind introduction. I'm going to start with the Inventor of the Year, and then we'll go on to the Transaction of the Year for 2021. So the Inventor of the Year award was created to recognize excellence in translational research, and specifically research that has, or that we believe will result in the development of a product that will benefit patients worldwide. This year, I'm pleased to announce that we're celebrating the contributions of 2 distinguished research teams, both of which I can say I thoroughly enjoyed working with, for their efforts in advancing technologies to combat SARS-CoV-2. These teams include members of the Center for Therapeutic Antibody Development and the Department of Microbiology. So I'm honored to recognize Dr. Andrew Duty, Dr. Thomas Kraus, Dr. Thomas Moran, and Dr. Domenico Tortorella as 2021 Inventors of the Year for the development of therapeutic humanized monoclonal antibodies against the spike protein of SARS-CoV-2 that have been shown to broadly neutralize the virus and inhibit infection in model systems. These antibodies have been licensed and are being developed commercially by Sorrento Therapeutics. So please join me in congratulating this team on their tremendous accomplishment.
Jacqueline, I think we have, yeah, there we go. So we had 2 winners this year, and maybe we can just ask any of the awardees if they want to say a word or two. Tom, Dom, or Andy?
Yeah, well, thank you, Scott. Thank you, Eric. I'd like to say a few words about the products and Mount Sinai community. The work that led to this award was a collaborative effort by the 4 of us in CTAD with a huge amount of support from Mount Sinai administration and faculty. While all 4 of us will not speak today, we are of one voice in specifically thanking all the entities that made these products possible. I want to particularly thank Dr. Charney and the Dean's Office, as well as Dr. Kenny and the Drug Discovery Institute for supporting the Center for Therapeutic Antibody Development, enabling us to take on these and other projects. In March 2020, we, like everyone else, were struck by the magnitude of the pandemic. In response, CTAD, with great efforts from Madhu Kumar and Mary Lopez in our center, began to develop products that are the basis of this award. Madhu and Mary continued to come to the lab every day and push forward our COVID projects at great personal risk. Our initial contribution to the efforts against COVID was the observation that monoclonal antibodies that we had made back in 2005 against the SARS-1 virus, along with Luis Martinez in the lab of Adolfo Garcia Sastre, were cross-reactive to the SARS-2 virus. We, with great help from the members of the micro department, showed that these antibodies could be used as reagents to identify the virus and monitor immunity. These antibodies were disseminated to all labs in Mount Sinai and eventually around the country for use as diagnostics. The value of these, while not as therapeutics, was nevertheless significant. The human antibodies for which we are receiving the awards were developed using humanized mice in our laboratory. We determined the specificity, binding affinity, viral neutralization of the antibodies using assays developed by us and some of our micro colleagues. The antibodies were shown to be competitive with any of the other therapeutics on the market, and importantly, were effective against many of the COVID mutant viruses that appeared. These efforts resulted in their successful licensing to Sorrento Therapeutics, as Eric has mentioned, for use as therapeutic monoclonal antibodies. Now I would like to introduce Professor Dom Tortorella, who will acknowledge members of the Mount Sinai community that contribute to this effort.
Great, Tom. Thanks. Just wanted to we would like to thank all members of the microbiology department, and specifically Dr. Peter Palese, Adolfo Garcia Sastra, Florian Kramer, Ben Hurley, Dr. Jeff Johnson, and Dr. Ana Fernandez Sesma for their generosity of reagents and expertise in coronavirus biology. Their labs are a hub of activity during the pandemic with a laser focus on gaining a comprehensive understanding of SARS-2 biology and developing SARS-2 therapeutics, vaccines, and diagnostics. We'd also like to give special thanks to the microbiology researchers and staff that were working in shifts, it seems like, during that pandemic. But these were the people who were in the trenches during the pandemic. From my lab, that was Sabrina Ophir, Andrea Parsons, as well as afterwards was Jaylene Paredes and Christina Tanisfoff. Other important contributions were from different labs from the microbiology department: Fatima Amatan, Lisa Miran, Soner Yildiz, Thomas Kerher, Wayna Sun, Shopei Agutuya, Christian Stevens, Satoshi Ikagama, and Richard Cadigan, who we worked really closely during the pandemic. Then finally, I would like to echo what Tom Moran, in which we are very grateful for the leadership at Mount Sinai and the commitment of the institution and the department for us to thrive in developing therapeutics against SARS-CoV-2, as well as future pathogens. So with that, we would like to, again, humbly thank you for this award. And I speak for all of us. We're looking forward to the next phases of development, as well as other opportunities to invent more things at Mount Sinai.
Thank you, Dom. Eric, would you like to introduce the other awardees?
Yes. Make sure that yep. So as I mentioned, we're honoring two groups, and you, as you just heard, there's tremendous collaboration between these groups. And again, as I mentioned earlier, it was phenomenal working with them and continuing to work with both. So I'm equally honored to recognize a second team for Inventors of the Year for 2021. And that includes Dr. Peter Palese, Dr. Adolfo Garcia Sastry, Dr. Florian Kramer, and Dr. Wina Sun for the development of NDV-HXPS, which is a novel vaccine against SARS-CoV-2 that utilizes an engineered version of the spike protein that is locked in the pre-fusion conformation. This vaccine has performed exceptionally well in human trials conducted in low and middle income countries in partnership with the global nonprofit PATH and a commercial licensee, and is advancing to later stages of development. We also believe that the vectors developed by this team may serve as the foundation for future additional vaccines with a potential for a greater global impact to assist low and middle income countries to become independent manufacturers of vaccines. So please join me in congratulating this team on their tremendous accomplishments for the development of NDV-HXPS.
Adolfo, I think you were slated to say a word or two.
Yes, so thanks a lot for this distinction. We are truly honored. Sorry, Peter Palese cannot be with us today, but I have with me our two co-inventors, Florian Kramer and Wina Sun. And when COVID-19 came and it was clear about the pandemic that was coming, then we got together, Wina, Florian, me, Peter, and asked ourselves, what could be the best contribution that we can do in order to do something for this thing that is coming? And then we thought, let's try a vector that we have been working for a while with, Newcastle disease virus vaccine vector. Let's try to move it quickly into clinical trials. It has a lot of big advantages that we could see, especially for low-income countries because of the easiness of manufacturing. But obviously, we need to start from scratch with respect to COVID-19 vaccines. We have some experience with Newcastle disease virus vaccines-based diseases, like other diseases like Ebola or RSV. We have good preclinical data, but COVID-19 was a completely new virus. So we got together, established a path, how to make the vector, how to move it forward. And it sounds easy, but it's extremely complicated. We needed to have serology in place to test the antibodies induced by the vectors that we will be making. We need to just have animal models that were not established at this moment in order to test our vaccines. We need to have partners to move this into clinical products, something that we have not done yet for Newcastle disease virus. And so although we are the ones that we are recognized for this invention, is with the help of a huge amount of help that we got from Mount Sinai, both with respect to funding, with the Dean's office, the people in the BSL-3 that are taking care of the BSL-3 facilities, engineers that expanded the BSL-3 facilities, select agent programs that we needed to use in order to do this thing safely. A lot of people contributed to that. GMP production. We needed the help from the people that they were doing GMP production. Nina, Barbas, Marcia, they were the ones that they were able to translate this thing into a clinical product, something that they would have not done it before. The serology needed to be put together in order to be able to test the antibodies induced by these vectors. And that was also a lot of contribution with multiple people like Carlos Cordon. So a lot of effort and obviously a lot of also funding that they came from Mount Sinai in order to move this forward. So we are now at the time in which we are conducting, together with the help of multiple governments in low and middle income countries, Thailand, Vietnam, Brazil, Mexico, also trying to do also clinical trials here with all the help of Mount Sinai, the people in the clinical, like Judy Aberg, that has been spending a lot of time in order to be able to move this forward for clinical trials here. We are now at the time that we are starting even phase 3 studies in one of the countries. So we hope that still we have time to be able to come out with a vaccine that will make an impact in places where still they cannot afford the vaccines that they have right now. But not only that, this effort has also validated the use of Newcastle disease viruses as vaccine platform. And we hope that we can continue with that and not only make an impact on COVID-19, but in other infectious diseases. So again, thanks a lot to the whole Mount Sinai family. It has not been like Tom Morancey and Dom Tortorella. It was only with the effort of everybody that we were able to have a program that was big enough to be able to move these things forward, which we cannot have done it alone by ourselves. Thanks a lot.
Well, thank you. So that obviously speaks to the tremendous collaborative environment at Mount Sinai. I would just like all of us to give a round of applause to the winners and frankly, to the whole Mount Sinai community. These are tremendous technologies. We continue to work on them and they are going to make a difference in patients' lives. So let's all give the winners a hand.
Okay, Eric, was there a transaction?
Yes, so there's, I'd like to move on to the transaction of the year. So the transaction of the year award was created to recognize Mount Sinai researchers or teams that have created technologies that have been licensed to commercial entities for development where the licensing transaction is a substantial and innovative transaction. And I'd say there's a lot of opportunities, or there's a lot of things that kind of meet that criteria, but one in particular this year stood out for us. And I'm honored to recognize again, Dr. Andrew Duty, Thomas Kraus, Thomas Moran, and Dom Tortorella, and the Center for Therapeutic Antibody Development for the 2021 transaction of the year, which includes the licensing of therapeutic humanized monoclonal antibodies to the spike protein of SARS-CoV-2 that I already described, but also a research collaboration with Sorrento Therapeutics for the development of additional therapeutic antibody candidates to benefit patients worldwide. So again, please join me in congratulating this team on their role in the 2021 transaction of the year.
So thank you, Eric. Technology management is an integral part of the discovery process. Without it, our efforts as inventor scientists would be worthless, really. And we have been incredibly lucky to not only be surrounded by great scientists and great thought leaders, purposely collected and collated here at Mount Sinai by our visionary leadership, who have created the foundations for great discoveries, but also by one of the most dynamic and capable offices of technology management in the academic sector. And that has made possible the execution of so many of these great ideas and inventions into actual practice. With that, our work in CTAD, the Center for Therapeutic Antibody Development, and the partnerships that we have instituted, very much not limited to our transaction this year with Sorrento Therapeutics, would not have been possible without our partners in discovery, and that's the Mount Sinai Innovation Partners, and specifically MSIP's President and Chief, Dr. Eric Liam, and the many, many affiliates there in the MSIP offices. We must specifically point out that we are overwhelmingly grateful for our MSIP director, Dr. Louise Lammers, who has been with us since the very beginning and has been our manager, our liaison, our counselor, and our constant supporter. She has helped build CTAD from its very beginnings to where we are today. And with that, we are very, very grateful, Louise. Thank you. Also, we would like to extend many, many thank yous to the office, to the larger offices of managers and directors and finance directors and legal teams in MSIP that have been instrumental in readying our assets for licensure, specifically Drs. Philippe Arejo, Christopher Friends, Drs. Patrim Patel, Patrick Chang. Also a special thank you to the legal teams, both patent and contractual, for all of their preparation, Sybil Lambillo, Phil Sembrivo, Tom Blankenship, Denise Welcott. And lastly, we are incredibly grateful for our industrial partners in this transaction, Sorrento Therapeutics, without which none of this would have been possible, for their continued work in preparing our monoclonal antibodies for the market. We especially would like to thank Sorrento's leadership, CEO Dr. Henry G, Senior Vice President for Antiviral and Immunotherapies, Dr. Robert Allen, and VP for Business Development and our business liaison, Bill Farley, as well as the many other scientists and directors of the company that have been instrumental in the further development of our anti-SARS portfolio. Thank you.
Thank you, Andy. So Eric, I'll pick it up from here.
Great, thank you so much.
I'm pleased to next introduce the Mount Sinai Pitch Challenge Award and to introduce the award and to reflect in many ways the breadth of what MSIP is doing. I'd like to introduce Cynthia Clayto. Before doing so, let me just say that one of the reasons I think Eric and I and others are averse to the term technology transfer is because that is a very, very narrow slice of what Eric and his team and Cynthia are doing. And this is one reflection of the creativeness with which they have educated our workforce and our scientists by creating opportunities to learn methods of entrepreneurship and to effectively exercise them, in this case, in the form of a pitch challenge. So with that, I'll introduce Cynthia Clayto, who's the Senior Director of Outreach, Entrepreneurship, and Special Programs of Mount Sinai Innovation Partners. Cynthia?
Thanks, Scott. Great. Great to be here, everybody. On behalf of Mount Sinai Innovation Partners, I'm pleased to present the Mount Sinai Pitch Challenge Award to this year's winner, Dr. Charles Mobbs. Charles invented a unique drug discovery platform that aims to treat age-related diseases with an initial focus on Alzheimer's. Charles is also the founder of the startup company Gilgamed, which serves as the basis for advancing this platform and its potential to impact patient lives. The Mount Sinai Pitch Challenge is an annual competition that serves as the culmination of MSIP's entrepreneurship program. The program helps evaluate, build, and refine healthcare solutions for Mount Sinai teams, and finalists who are accepted into the Pitch Challenge compete for a cash prize and pro bono services to continue advancing their solutions towards commercialization. Since the Pitch Challenge, which took place on May 5th this year, Gilgamed has put the prize money to good use by advancing its technology, expanding customer discovery efforts, and financing strategies, and are in the process of joining external accelerators. MSIP looks forward to Gilgamed's next steps towards commercialization and its potential to impact age-related diseases. Congratulations to Charles on this exciting achievement.
Wow, that's quite an intro. I don't have a lot more to add to that, but I do want to express my appreciation, though, and actually delight for this acknowledgement, as well as just the program itself, which I think is a good example of the many terrific innovations that MSIP has been developing. So I just want to sort of raise a question, because why does a person want to become a scientist? Compared to the long years of education it requires, it does not pay particularly well, isn't that prestigious. And in fact, unfortunately, as we know, a lot of people are now hostile to science and scientists. And it's the very rare scientist who is nominated for the sexiest person of the year. Of course, Dr. Fauci is an exception. So what's the appeal? Well, curiosity is an almost universal trait in the animal kingdom. And for some people, curiosity is kind of their main personality trait. And I have to say that's true for me. So for those that are driven by curiosity, science is the best way to go. Okay, so scientists are driven by curiosity. So then the question is, what questions do you want to spend your life studying? I realized that curiosity wasn't the whole thing for me. I also kind of wanted to save the world. And so there's a lot of very interesting science out there, but I kind of had the feeling that biomedical research was going to be my best chance at saving the world, I guess, doing the most good. So I decided to make aging and age-related diseases the focus of my research program. Especially what's the link between age and age-related diseases, and how dietary restriction delays all age-related diseases. Because the plan was to figure out how that works and put it in a pill. And thanks to Mount Sinai and MSIP and my many other colleagues at Mount Sinai, we're actually surprisingly close to developing such therapies for Alzheimer's disease and a wide variety of other age-related diseases. So there's so many people at Mount Sinai I really want to thank. I can't thank everyone, but I'm going to pick out a few people especially. I especially, again, want to thank MSIP. None of this could have happened without you folks, especially Dr. Louise Lammers and the 4D program, which funded the initial screens that made all this possible. And of course, Dr. Dov Shamir, who has been like so absolutely critical at every step of the way, from our initial discoveries to starting the biotech company. And the biotech company was there and encouraged by MSIP, because that's the best way to develop the resources that's going to be necessary to take our drug discovery into the clinic. Obviously, I want to thank the members of my lab, especially Dr. Rachel Litke, who carried out the initial screens, phenotypic screens funded by the 4D program that led to our present highly promising drugs, which inhibit both A-beta toxicity and microglial TNF-alpha release, both implicated in driving Alzheimer's disease and many other age-related disease, and mimic the protective effects of dietary restriction to increase lifespan. James Fikari and Bixu Kwong greatly extended those results in a variety of initial screens. Dr. John Jen oversaw the synthesis of our novel compositions of matter, patent pending, thanks to MSIP. Dr. Chris Kellner, who supervised the studies demonstrating these drugs also protect in a mouse model of stroke, and especially Dr. Paul Kenny and Dr. Scott Friedman, who have just been the most amazing sources of advice and moral support, which I need a lot of during all of these studies. So again, thank you. And I apologize for the many other people I just don't have time to thank. Thank you.
Congratulations, Charles. Based on your background, I think at least today you're the king of New York.
Yes, I like to think so.
Congratulations.
Thank you.
Next, I'd like to introduce Dr. Emma Ben, who will introduce the Faculty Idea Prize awardees. Dr. Ben is an associate professor in the Center for Biostatistics and Department of Population Health Science and Policy, and the founding director of the Center for Scientific Diversity at the Icahn School of Medicine at Mount Sinai. Emma?
Thank you. So I am presenting this award in my role, my additional role, Associate Dean of Faculty Wellbeing and Development. The Office of Faculty Development believes that high quality, enriched mentorship and leadership training are vital approaches to fostering faculty career development and well-being. The Faculty Innovative Idea Prize is one of our methods to support the research careers of junior faculty through pilot funding. The review committee is very proud to present the 2021 award to a team of truly inspiring junior faculty. Dr. Sarah Miller, Jamelia Sly, Tracy Lane, and Urena Clark have proposed to develop and implement a digital platform called PAIRED, which stands for the Portal for Advancing Inclusion, Research Excellence, and Diversity. The goal of this platform is to match underrepresented trainees interested in pursuing biomedical research with NIH-funded PIs, principal investigators at Mount Sinai, and ultimately scaling PAIRED up to optimize its impact on a national level. As leaders in the Center for Scientific Diversity at Mount Sinai, this team will support faculty mentors to increase their likelihood of submitting a successful NIH diversity supplement and ensuring that they apply an anti-racist, inclusive, and equitable approach to mentoring. Please join the Office of Faculty Development in congratulating this excellent team of investigators.
So Sarah Miller, would you like to say a few words?
Yes. Thank you so much for this award. On behalf of Dr. Sly, Dr. Clark, Dr. Lane, and myself, we are really honored to receive this award. As Dr. Ben explained, this award will allow us to build and pilot test the digital portal for advancing inclusion, research excellence, and diversity, or PAIRED. And the PAIRED portal is designed to match trainees who are underrepresented in science with NIH-funded scientists, hoping that we can help them apply for NIH diversity supplements. And we will also, the platform will also provide resources for mentors to ensure that they apply an anti-racist and inclusive approach to their mentoring, and that is at the foundation of the work that they're doing. So we hope that this platform can be utilized to increase training opportunities for individuals who are underrepresented in science, with the ultimate goal of promoting inclusive excellence. This project really represents a collaborative team effort within the Center for Scientific Diversity, and it's something that we've been talking about and wanting to work on for about a year now, and we are truly excited to begin this work. So thank you very much.
Congratulations to you and the team. It's a wonderful idea. I also want to give a shout out to Lakshmi Devi, who in her prior role in the Office of Faculty Wellbeing and Development had conceived of this prize, and she was involved along with Eric Liam and I and others in choosing the winner. So congratulations, wonderful prize and winners. Next, I'd like to introduce Dr. Ilse Dunn, who will present the Killen Postdoctoral Entrepreneurship Award. Ilse is an associate professor of medicine in the Division of Nephrology. She and her colleagues, when they were postdocs, and I think you'll tell the story better than me, Ilse, but what was remarkable is they had funding available, and their first impulse was to give it back to Mount Sinai in the form of this award. So we're very grateful both for the award and also for the leadership in showing loyalty and commitment to our own scientific community. Ilse?
Thank you, Scott. And it is with great pleasure that I present the Keystone for Incubating Innovation in Life Science Network, Killen Award, to recognize and support entrepreneurial trainees in the Mount Sinai Health System. With Doctors Nicole McKnight, Marina Verghese, Gabriela Casalena, Yana Zorina, and myself, we founded Killen while we were postdocs here at Mount Sinai with the goal to build a culture that sustains innovation in life sciences by providing programming to entrepreneurs in New York City. This is the third year that the award's been presented, recognizing postdoc fellows from our community who co-found or lead companies that address unmet needs in life sciences, including but not limited to basic or translational science, clinical care delivery, and that they take these discoveries beyond the bench. Past winners include Raymond Alvarez, who's CEO of Icor Biologics, who is advancing his platform technology for developing human monoclonal antibody-based therapies. Last year, Carlos Felipe Pereira, CEO of Asgard Therapeutics, was awarded, and he announced last week that they won the Eureka Eurostar grant for 2 million euros. So this is fantastic, and I am delighted today to present this year's winner of the ISMMS and Kiln Postdoctoral Entrepreneurial Award to Dr. Philip Muse, co-founder of Epivario, a biotech startup focused on novel approach for treating PTSD. Please join me in congratulating Dr. Muse.
Thank you so much. Yeah, thank you so much to Kiln, to the selection committee, and really to all of those making this prize possible. This is an incredible honor. And yeah, my co-founder, Dr. Shelley Berger, and I, we launched Epivario to bring a new treatment to people with post-traumatic stress disorder to diminish traumatic memories. And when we started pitching our idea and how we would, in fact, accomplish that, we started talking about the eternal sunshine of a spotless mind, which people understood immediately, but they're also freaked out. And so we had to rework the pitch a little bit, but we truly think that we are close to a new treatment for PTSD, which is really an insidious, debilitating disease that continues to be fatal every day. And yeah, the fact that this prize has come out of Sinai is really indicative of the amazing connections in Mount Sinai network, in particular, Dr. Rachel Yehuda and Dr. Eric Nestler, who are important advisors to the company. And I also want to thank our fantastic collaborators at Sinai, in particular, Dr. Paul Kenny. And so, yeah, we are really incredibly thankful to the Kiln Prize for recognizing our work and for being a part of our solution for PTSD and also addiction. And I would have never thought that my PhD work would evolve into this company. And so the visibility that this brings to us will hopefully will bring more partnerships and accelerate our growth. And I want to say thank you again. We are very grateful for this prize.
Congratulations again, and thank you again, Ilse, to you and your colleagues for your commitment to this program. Next, it's my pleasure to introduce Dr. Janice Gabrilov. Janice is the Director of Clinical Research Education in the Graduate School of Biomedical Sciences and Associate Director of Education and Training in the Tisch Cancer Institute. Just by way of a little bit of history here, this fellowship was created in the context of the COVID pandemic. As some of you may recall, we developed a very successful hackathon program that accompanied our Sinai Innovations event each year. But with the canceling of an in-person event, Janice and her team pivoted remarkably quickly and created or conceived of this fellowship that in many ways expands the vision of the hackathon through an entire year. It's been wildly successful, extremely creative. So I want to thank you, Janice, and your team, and allow you to introduce this year's winner.
Thank you so much, Scott, for those kind remarks. It gives me tremendous pleasure and an honor to present the awards for the Thrive Fellowship. Thrive is Targeted Healthcare Innovation Fellowship, and I'm presenting this on behalf of my colleagues who are part of the Thrive Organizing Committee. Thrive is a 9-month program designed to catalyze health tech innovation in response to needs arising from the COVID-19 pandemic, and was meant to encourage collaboration among the participants from diverse scientific backgrounds and perspectives across the medical school, the graduate school, and the Mount Sinai Health System at large. It was intended to propel experiential team science training and skill building while accelerating the process of ideation and development of commercially viable prototypes. It has a structure of guided mentorship from top researchers here at Mount Sinai, and we're fortunate to be able to have our participants engage in the MSIP Entrepreneurship Bootcamp as part of their curricular efforts. It is enrolled through a competitive application process and is really the brainchild and represents a partnership of Sinai Biodesign and Conduits, with additional generous support and vision from the Office of the Dean. Our first recipients is Virtu, and their product is Normal. The invention is a tool employing machine learning technology to address infection prevention needs for social gatherings. This team has been remarkably productive, and its accomplishments include placing third in the MSIP showcase competition that we just heard about. They participated in the New York City business plan competition and came in first in the infrastructure, first responder, and defense track. They advanced to the New York State Business Plan Competition Awards, again in the infrastructure track, and the Foos Hub Pandemic Response Award. And finally, they've participated in the New York City Regional Innovation Node of the NSF I-Corps program. I'd like to indicate that this team represents the collaboration that we really looked for with team members including Gary Gravesandi, a candidate for MD in his second year, Sophia Salazar, a doctoral candidate here in the graduate school, Tafane Martin, a PhD and master's in engineering, Janki Shah, a physician, and Jerry Sun, a master's in healthcare administration. Diverse teams working together for a really fantastic prototype well on its way. Sophia, would you like to say a few comments?
Of course. Thank you. Thank you, Dr. Gavrilov. Well, so far, only 3 of us have actually met in person. And thanks to our tech, this is the first time that we're actually in the same room. So as you can say, it's taken a bit of creativity to develop an idea and to work as a team to accomplish what we have in the past year. So to surmise our technology, we were designing a platform that would harness the power of machine learning technology to address infection prevention for large social gatherings. In short, we were taking an intelligent approach to work and to actually make it an intelligent approach to actually getting back to normal. So after winning a few of the entrepreneurship competitions that Dr. Gavrilov mentioned, and having the successes that we've had so far, we'd like to take the opportunity to thank everyone who have put in the time and the effort despite the challenges that the pandemic brought upon us. So this is going to be quite a list because a lot of people took turns into mentoring and guiding us throughout our journey. So we wanted to start with our Thrive program leaders, Dr. Gavrilov, Layla, Joe, Anthony, our Thrive mentors, Dr. Emanuela Talley, Dr. Chris Kellner, and Dr. David Petrino, as well as all the guest speakers throughout the fellowship that really inspired us to come up with the idea in the first place. Our mentors from the NSF Regional I-Corps, Ariella, Arbor, and Jessica, as well as Nitran, who basically facilitated all the communications with everyone. And then MSIP leadership and the mentors that we gained from them, Dove, Karady, Ruben, and our MSIP fellows, MD, and Divya. Other faculty members that also motivated us to pursue entrepreneurship endeavors, including Dr. Pricker and Dr. Nickerson, the Channel Studios team who helped bring this beautiful app design to life, and actually our working model. So that is Seth, Will, and Maori. And last but certainly not least, our PIs who lent us the bandwidth to pursue this endeavor. My PI, David Petrino, and Tifane's PI, Dr. Parsons, as well as Janki's advisor, Dr. Iloretta. We also wish we could say more for each and every person, but we only have 3 minutes. So we hope that someday soon we could personally express our gratitude for your contribution and share more future successes with you. If only we could have convinced our competition judges to believe in the longevity of our product as much as our mentors did, we may have had this award ceremony in person. But nevertheless, we thank you all for your recognition and the wonderful opportunities that this has brought us to this day. So thank you.
So our second team to be recognized today is Team Change Detection. This is a team that developed a New York City syndromic surveillance system utilizing machine learning analysis of real-time patient data. It's meant to detect deviations in emergency room arrivals in terms of patient case mix, disease distribution, and severity index. And they work to train and test a Python algorithm using New York City COVID-19 data. This type of approach of trying to anticipate where surges may be occurring has implications also for early detection of outbreaks in a more static population where there is less noise apparent. Our team, again, for Team Change Detection, brings together 4 talented, diverse individuals. Tina Chen, who is a doctoral candidate for a PhD in clinical research. Olivia Cullen, who is a candidate for a master's in clinical research. Joy Jiang, who's an MD PhD candidate. And Katie Link, a candidate for MD, currently in her 2nd year. Olivia, would you care to say a few comments, please?
Yeah, so hello everyone, and thank you, Dr. Gabrilov, for the introduction, and to everyone at Thrive, like such a thank you for the tremendous help along the way. So when my team and I applied to the Thrive Fellowship back in 2020, our ultimate goal was to help address pandemic-related issues. Following an amazing lecture from data scientist Dr. Beth Percha, we became curious to affirm if COVID-19 could have been detected in New York City via emergency room admissions, like long before New York had to declare a state of emergency. Joining Dr. Percha, as well as Dr. Alexis Dabrowski and Max Tomlinson, our team of Thrive researchers, including Joy Jiang, Tina Chen, Katie Link, and myself, formulated a project to address this research question. As was mentioned, the project focused on the construction of a New York City syndromic surveillance system, which basically involves the analysis of real-time patient data via a machine learning algorithm to detect potential infectious disease outbreaks. We wanted to create a system that would alert public health officials concerning any unexpected or unusual health system activity that might indicate the presence of a novel disease or some type of outbreak. And so with earlier detection, public health officials would thereby be able to investigate these abnormalities more quickly, enabling them to mobilize resources and minimize potential fallout. This project helped us to better understand and continue to explore the variety of applications for this type of algorithm moving forward. So thank you so much for this award, and I'm honored to accept it on behalf of my team.
Thank you.
This
Scott,
award,
back to you.
Back to me? Okay. What's remarkable, as trite as it may sound, is necessity was certainly the mother of invention when it came to COVID and the response of so many of the people being recognized here. So I'd like to move on now and introduce Margaret Barron, MD PhD, who's the Senior Associate Dean for MD PhD Education and Director of the MD PhD program. And she's also a professor in medicine in the Division of Hematology and the Department of Cell Development and Regenerative Biology. And she in turn will introduce and recognize the winner of the MD and MD PhD Student Trainee Innovation Idea Award. Margaret?
Thank you, Scott. It's a very great pleasure to present this Trainee Innovation Award to Nima Assad, an MD PhD student at Mount Sinai, for his team's proposal, Spatial Immune Transcriptome Visualization in Human Tumors. This work is a tour de force in utilizing state-of-the-art technologies to address the huge unmet need for effective therapy of liver cancer. It will help us understand the high degree of heterogeneity in liver cancer and could lead to the development of better treatments for this disease. Nima.
Thank you, Dr. Barron. It's humbling to be on a panel with so many scientists that I admire and I look up to. And it's my pleasure to accept this award on behalf of the entire lab of Dr. Miriam Murad at the Precision Immunology Institute at Mount Sinai. So we've long been interested in understanding the immune response to tumors, and now with advanced sequencing techniques such as spatial transcriptomics, we're able to ask and to address questions which we've never been able to. So I have the privilege of being a second-year MD PhD student at Mount Sinai, which has given me the resources and mentorship to develop ideas such as the quantification and visualization of immune aggregates in human tumors. I would like to thank the organizers, administrators, and leadership at Mount Sinai and within our department. the generous donors to PRISM, our Precision Immunology Institute, and our industry partners who have all contributed to make access to cutting edge platforms such as spatial transcriptomics possible for us. And finally, a thank you to our patients who have donated tissue sample for us to study, and a promise on behalf of us as scientists to develop the next generation of therapies for cancer. Thank you.
Thank you, Nima. Congratulations to you and your team. Even though I study liver and liver cancer, I want you to know I had no bias in assessing the excellence of your award. Congratulations. Great stuff. Now it gives me pleasure to introduce Matthew O'Connell. Dr. O'Connell is a Senior Associate Dean for Curriculum Recruitment and Admissions in the Graduate School and a Professor of Oncological Sciences. Matthew?
Thank you, Scott. It is quite an honor to be a member of an institution with all this incredible research and innovation that we're hearing about today. And this also speaks to the volumes of outstanding research that's being done by our graduate students in the graduate school. They study all sorts of areas of biomedical research, from the very basic through to the very translational, and are making cutting edge breakthroughs on a daily basis. For our award today, we are here to honor Yi-Yang Chen, a PhD student who works in the cancer biology program in Dr. Jerry Chippick's lab. What Yi-Yang is being recognized for today is his outstanding work in developing an immunohistochemical-based prognostic screen to better manage the very deadly disease of malignant melanoma. So my congratulations to you, Yi-Yang, for your tremendous work.
Thanks, Matthew, for these words, and I really feel delighted to receive this innovation award. This project is about generating a recombinant antibody as a biomarker, which can be used in the prognostic IHC staining for early detection of melanoma, as well as monitoring melanoma progression. This IHC-based assay is simple, accessible, and will potentially reduce the healthcare cost while maintaining accuracy in the evaluation. And I'd like to thank the program committee, the program organization, and also Mount Sinai for this Innovation Idea Award. I'd like to thank the Dr. Simon Siga and also Dr. Wade Wider, whose research laid the foundation for my project. I also want to thank Dr. Chip Lab. We are a unique, resilient group that love each other, collaborate each other, and thrive together. My PhD journey is much more joyful because of you guys. And lastly, I want to thank my PI, Jared Chiphook, for his diligent supervision and care in my PhD student study. Thanks, everyone. Again, I feel really honored to receive this award. Thanks.
Congratulations, Yi-Yang, and also a shout out to Matthew, who's been just a wonderful leader and role model in our educational program. It's really a pleasure to work with you, Matthew, and congratulations again, Yi-Yang. And then the final presenter, last but not least, is Dr. Mike Lightman, who's the Dean for Graduate Medical Education and a Professor of Surgery and Professor of Medical Education. Mike oversees the largest training program cohort in the United States. He does it with enormous grace and wisdom and balance. And so it's a pleasure to introduce you to have us hear about the last two awardees, Mike.
Well, thank you, Scott. It's really an honor for me to join you in recognizing the exceptional work of our postdoctoral fellows and our residents and clinical fellows. Their accomplishments under the tremendous weight of this global pandemic are even more notable. The first award goes to a postdoctoral fellow on behalf of Dean Marta Villazola and Dr. Guy Montgomery from the Graduate School of Biomedical Sciences, and it is a COVID-related project. Dr. Alba Boix and her collaborator, Dr. Jose Clemente. Dr. Boix arrived at Mount Sinai on the very eve of the pandemic in January 2020, and her project was entitled an in vitro model to study the role of nasopharyngeal microbiome in SARS-CoV-2 infections in a low biosafety environment. Congratulations to Dr. Boix and Dr. Clemente.
Thank you so much. Thanks for the award. So basically, our idea consists of developing an in vitro cell model that reproduces infection by SARS-CoV-2 and allows to test whether commensal bacteria naturally present in the nasopharyngeal tract
might reduce the binding capabilities of the virus to its receptor on host cells. And as Michael said, our model allows the study of viral host interactions, but does not require the presence of the complete virus, which eliminates the need for working in high biosafety level containment capabilities, BSL-3 labs, which we all know can be very challenging for researchers. And I just wanted to say thank you to Mount Sinai Innovations for organizing the event and for selecting my idea. Of course, my PI, Jose Clemente, for supporting me and for encouraging me to develop the project and to apply for the award. Everyone else in my lab, of course, for the emotional support, especially to Enrica Piras. She's no longer with us, but she helped me draft this idea back in the first months of the pandemic. And finally, to Glenn Doherty from the microscopy core for being so helpful, so patient and kind with me, helping me to set up the microscopy part for the analysis of this project. So thank you so much.
Congratulations again. Our final award goes to Megan Januska from the Department of Pediatrics. She and her co-investigators, Yafei Sun and Martin Walsh, their project was entitled Flexible Bronchoscopy and Single Cell RNA Sequencing, a new workflow for personalizing cystic fibrosis care. Dr. Januska is a third-year pediatric pulmonary fellow at the Kravis Children's Hospital and is a graduate of the Dartmouth School of Medicine and our pediatric residency program at Kravis, Mount Sinai. Dr. Januska has also since secured a 2-year Junior Investigator Award from the Cystic Fibrosis Foundation. Congratulations, Dr. Januska.
Thank you, and thank you for that introduction and for this award. So outside of the realms of COVID-19, cystic fibrosis, or CF, is a multisystemic autosomal recessive disorder that's caused by variants in the CFTR gene, with the majority of morbidity and mortality extending from lung disease. And while the 21st century has seen tremendous advancements in the care of people with CF, particularly with the advent of CFTR protein modulator therapies like Trikafta, the effect of these advancements has not been uniformly expressed throughout the CF community, which really has provided the impetus for this work, providing care for those who can't access these therapies. We know that disease-related changes in the CF airway have been observed in explanted lung tissue from persons undergoing lung transplant through single-cell RNA sequencing. And our group really aims to develop a minimally invasive workflow, profiling bronchoalveolar lavage fluid samples and tracheobronchial brush biopsies acquired during flexible bronchoscopy, a minimally invasive procedure, to detect those disease-related changes earlier to enable a more refined method to monitor disease progression, tailor treatment, and determine treatment efficacy. Robust characterization of a single cell cohort in the CF airway, which we are currently building, will establish a valuable resource for future investigation, allowing us to present targets and opportunities for reprogramming specific respiratory epithelial cell types, offering novel approaches to CF therapeutics, as well as clinical translation, detecting disease-related changes in the CF airway over time with disease progression and after intervention. So in addition to my brilliant team, Yafei Sun and Marty Walsh, who have been very patient with my learning curve, I would like to thank Kristen Beaumont and Bobby Sebra and their teams in the Mount Sinai Genomics Core facility for their assistance and contributions with this project, as well as Didi Vicencio for his ever-present support and guidance. Thank you all.
Congratulations again, Megan, and thank you, Mike. So this comes to the end of our ceremony, but I'm sure it's just the beginning of greatness for many of those who've been recognized today and others like them throughout the Mount Sinai community. For me, it's been an absolute privilege to represent and reward so much extraordinary creativity through these awards, but equally through all the day-to-day efforts that contribute to an ecosystem that is unparalleled in its ability to drive innovation, promote entrepreneurship, and ultimately improve the lives of our patients, as so nicely and importantly articulated by Dean Charney. So I want to just ask you all to give one more round of applause to all our awardees and thank the presenters again. Congratulations, and we'll see you all next year, hopefully in person. But regardless, I'm sure that the spark of creativity that is epitomized by these awards will continue to grow. Thank you all and good night.